vaccines. And so essentially what we're doing here is going back to our pathogen. We're saying, okay, look, we know that there's specific proteins on some of these pathogens that we can isolate. Instead of giving this patient the whole organism, let's just give them maybe a couple of those proteins. Okay, so maybe we just give them a couple of those proteins. And these proteins, we can then target these proteins and make antibodies against them. Okay, so we can take these antigens from this species, right, whatever it is that was on the cell surface, and we can make antibodies from that. All right, and so that's the concept. So we're just giving the protein components of the pathogen. So we wouldn't expect there to be any issues with, a replicating live pathogen or anything like that, because I'm just giving the proteins off of that virus's cell surface, for example, or from that bacteria. And again, you might be saying, okay, well, what kind of immune response are we going to generate here? And we can see it's humoral IgG. Why is that? Well, remember, if I have this antigen floating around in the blood, I can have a dendritic cell come up to that antigen. It can digest that antigen. Okay, so it can digest that antigen, and then that antigen can get displayed in MHC class 2 because I phagocytosed it with my antigen presenting cell, and then eventually lead to the formation of antibodies, IgG antibodies from class switching. It's the same process we keep talking about. And again, you might say, well, was there a cell-mediated response from this? And the answer is no, right? There's not going to be one for the same reason we talked about when we talked about the whole inactivated vaccines, because this antigen is not a live organism infecting cells, right? It's not going to infect a dendritic cell. It's just a protein component floating around in the serum that we're generating antibodies to and phagocytosing. Okay, so for that reason, you're not going to get any cell-mediated response again with a subunit protein inactivated vaccine, right? You're just giving this patient a protein or an antigen from that pathogen. So you're only going to get humoral IgG response. If we're stratifying this is going to be in the middle somewhere in terms of its immune response. Pertussis is very classic. And then we also have hepatitis B, right? We talked about that a little bit in the last video. Hep B surface antigen, and we'll talk about this in greater detail when we talk about the microbiology section, but the Hep B surface antigen is going to be what we're targeting. So again, it's just like it says, right? If I have some virus here, if this is my Hep B virus, okay, I'll just put HB here for my Hep B virus, and on that Hep B virus, there's going to be certain surface antigens, okay? And instead of giving the patient a vaccine that has the whole Hep B virus, I might just give them the antigen, right? I might just give them what the antibody targets, for example. Okay, and then we can also see HPV is on here, and then toxoid vaccines. And what we're doing is we're essentially giving the patient this toxin, but its pathogenicity has been wiped, right? So it's not going to be causing disease, but it still has immunogenicity. Okay, so I can still make antibodies to it. So pathogenicity down, immunogenicity, we can still generate antibodies to, and that's the concept. Okay, so that's what we'll see with tetanus and diphtheria. And sometimes you'll see this vaccine, right? This is going to be a very classic vaccine, especially when you get to step 2. You'll see the Tdap vaccine, right? Tdap. So what does this stand for? So the T is for tetanus, right? That we just saw. Okay, so that uses a toxoid. Then we saw the D here. This is for diphtheria. Okay, and what is this AP? AP is what we have right here, right? AP is acellular pertussis. Acellular pertussis. Why does it say acellular? Because the organism is not being given. We're just giving the subunit protein with 2 toxoids.